Hello students, the first lecture for today will be glycogen. Let's see what is this. Where is a glycogen? Glycogen, students, is located actually everywhere in all cells, mainly in the liver and muscle tissues, but is located in all cells. Why? Because glycogen is the main storage carbohydrate for us. Actually, we have two storages for us. The first one is glycogen from the carbohydrates, and the second one is TAG, triacylglycerol, from lipids. But we don't have proteins as storage. Sometimes during long-term starvation you can use muscle proteins, but only during the starvation. And the main carbohydrate from the food is a starch. Starch is the main storage polysaccharide in plants, and in a major carbohydrate source is the human diet. It consists of a mixture of two components, amylose, which is a linear molecule composed of glucose residues, and amylopectin, which is a branched polymer similar to glycogen with less frequent branches. The biological significance of glycogen is that after the combination of these glucose molecules linearly or by branches, there is decrease the osmotic pressure of the cell. It's absolutely the same like in the formation of TAG, it's the same. Thanks to formation of TAG or glycogen, there is decrease of the osmotic pressure of these storage forms. Hydrolytic degradation of glycogen and starch. The process starts in the digestive tract by the action of amylases. You know two amylases, salivary amylase, alpha amylase, and pancreatic amylase, which is secreted by the pancreas, the exocrine pancreas, and the action is in the small intestines. And they hydrolyze alpha-1,4 glycosidic bonds within the polysaccharide chain, and the second enzyme which is used is a so-called debranchic enzyme. Debranchic enzyme is used for the degradation of both alpha-1,4 and alpha-1,6 glycosidic bonds at the branching point. And after that, after the full degradation of starch, are produced in the end disaccharidases, and after disaccharidases are produced monosaccharides, predominantly glucose and also fructose and galactose in the end. And this is the structure of glycogen. Glycogen structure, many glucose molecules. The linear chain is thanks to these bonds, which are called alpha-1,4 glycosidic bonds, and the places of branches, the bonds are called alpha-1,6 glycosidic bonds. In the structure of glycogen, students, there is one reducing end with free OH group at the position C1, carbon number 1, and many non-reducing ends with free hydroxyl group at the position 4, C4. It's important to understand. About the liver and skeletal muscle, the liver in humans, glycogen is about 10% of its weight. In the skeletal muscle, about 1–2% of their weight. Glycogen serves as a rapid source of glucose when we need it. When we need glycogenolysis, students? Well, when? To answer this question, tell me before that, how can we maintain the glucose in the blood? First, by eating of carbohydrate food. Second, liver glycogenolysis, which means if we don't have carbohydrate food eating, and if there is more than 3–4 hours without carbohydrate food, start the glycogenolysis in the liver and can maintain the glucose in the blood around to 18, not more than 18–20 hours, because after that start the third process for the maintain the glucose in the blood, which is called glycogenogenesis. Yes. We start with glycogen degradation, but we're talking about the intracellular degradation of glycogen, which is called glycogenolysis. The main enzyme that you know from the first colloquium is called glycogen phosphorylase. This enzyme is the first one. The second one is called the branching enzyme, with two activities you see, and the third enzyme is called phosphoglucomutase. Let's see the scheme directly to understand the process. The process, students, first we need to explain when the process will be active. I mean, during, we said, but let's repeat again, during the starvation state or during the well-fed state? The starvation, yes. But if we have starvation state, which is the hormone? Glucagon. The hormone of starvation, actually they can be all contrainsular hormones, but the hormone of starvation is glucagon. The hormone of stress is adrenaline and glucagon and cortisol. Cortisol action is in the muscle proteolysis. Adrenaline action can be in the liver and muscles, but the glucagon action is only where, students? Which organ is the place of action of glucagon? Liver. Why not muscles? Because they don't have glucagon receptors and they don't have the enzyme glucose 6-phosphatase. That's why. Okay, let's start with glucagon, adrenaline, etc. They activate the enzyme glycogen phosphorylase because they activate which enzyme from the reversible covalent modification? Called kinases, and that's why glycogen phosphorylase becomes active in its phosphorylated form. Yes, and the process starts. Start from non-reducing ends, and start phosphorylitic degradation thanks to inorganic phosphate, which is contacted with each of glucose molecules one by one. Phosphate enters, glucose 1-phosphate is released. Phosphate enters, glucose 1-phosphate is released. The product is glucose 1-phosphate, is not free glucose. In the 90% of situations, the glycogenolysis is by this way, by glycogen phosphorylase and glucose 1-phosphate production. But when we're talking about maintaining the blood glucose level, we don't talking about phosphorylated glucose molecules. We mean free glucose. That's why we need more enzymes. The next one is called phosphoglucomutase to produce glucose 6-phosphate, and after that, thanks to this enzyme glucose 6-phosphatase, which is located in the liver and kidneys. These are both of the organs of glucogenogenesis, by the way, because this enzyme is a part of glucogenogenesis, glucose 6-phosphatase. That's why free glucose will be produced in the end. With other words, 90% of the production of free glucose is thanks to this way. And this enzyme is active when 4 or 5 glucose molecules remain to the branch and stop the action of glycogen phosphorylase. 4 or 5 glucose molecules to the branch. Because glycogen phosphorylase is active only in the degradation of alpha-1,4 glycosidic bonds, but not alpha-1,6. That's why I start the action of the next enzyme, which is called debranching enzyme. Debranching enzyme has 2 activities. The first activity is called glucan transferase activity, which means catches 3-4 glucose molecules from the branch without the last molecule, without the last molecule. The last molecule remains here because this molecule is connected with 1,6 bond without this one. And after that, this branch is moved to the linear chain, and the second activity of the branching enzyme called amylo-1,6-glucosidase is used for the hydrolysis and releasing of this glucose molecule as free one. 10% of glycogenolysis is happened by this way, thanks to the branching enzyme, which means if there is anywhere before that deficiencies in glycogen phosphorylase or phosphoglucomutase or glucose 6-phosphatase, no possibility to produce glucose by this 90%, which means in these situations the organism will rely on only the branching enzyme action. 10%, 10%. And something about the glycogen phosphorylase enzyme. The enzyme acts by the degradation, this is the exoenzyme, because the degradation is by this mechanism from non-reducing ends of glycogen removes these glucose molecules. And the enzyme is dimer composed of 2 identical subunits. Okay, we said about that. We said about this enzyme, and here are this is the metabolic fate of glucose 1-phosphate. Actually, you know the scheme from previous lectures. Phosphoglucomutase is a reversible reaction. This reaction is reversible, and glucose 6-phosphate is a key metabolite, which can be directed to glycogenogenesis to produce glucose in the liver when we need glucose, or can be directed to some breakdown pathways of glucose. There are 2 breakdown pathways of glucose. The first main one is glycolysis, and the second alternative pathway is called PPP. It depends on the cell need, where will be the direction. The next process, opposite one, is glycogen synthesis. But glycogen synthesis and glycogen degradation are not so opposite enzymes that you know, like glycolysis and gluconeogenesis. Because in glycolysis and gluconeogenesis, they are almost the same enzyme, with the exceptions of these 3 irreversible reactions. But here are absolutely different enzymes. Let's see the process. Actually, the main enzymes involved in glycogen synthesis are these 5 enzymes: hexokinase in muscles or glucokinase in the liver, phosphoglucomutase again, UDP glucose pyrophosphorylase, tyrosine glycosyl transferase, and glycogen synthase, which is the main one, the main active enzyme. Let's see the first process called activation. Students, I will remind you again that all processes, never mind where will be the direction, need to have active substrates molecules. The substrates need to be activated in advance before that. And here there is one twice activation. There is double step of activation of glucose. There is the first activation is hexokinase reaction. Here is written glucokinase if you're talking about the liver. And glucose 6-phosphate is a more active compound than glucose. Why? Because it has more energy than the glucose. The next activation, this is the second activation, is using UDP glucose pyrophosphorylase and 1 molecule UTP to produce UDP glucose. Actually, you know about UDP glucose from this week when we talked about lactose synthesis from glucose. There was the same situation was with the production of UDP glucose again. This is the double step activation. Why? Because there is first we lose 1 ATP, and second we lose 1 UTP. 2 molecules we lose for the formation of this more active substrate called UDP glucose. That's why when we ask which is the substrate for glycogen synthesis, okay, it's a glucose, but the active form is UDP glucose. This is the real substrate for glycogen synthesis. After that, the second process is formation of a glycogen primer. This primer is required, which has short oligosaccharide chain with around 7 glucose molecules, and in muscle tissue glycogenin remains in the core. In the liver can be released and reused in another glycogen synthesis. Let's see again the scheme. You see all these enzymes. Let's start from the beginning. The beginning is formation of active form of glucose called UDP glucose. We start again from hexokinase, phosphoglucosamidase, and UDP glucose is ready. After that, students, there is involvement of one primer which is called glycogenin. Glycogenin is one molecule which has autocatalytic functions and can attach around 7 glucose molecules to its hydroxyl group to form one small chain with 7-8 glucose molecules, which is called glycogenin with glycogen primer. And this is the beginning. After that, sometimes in other cells can be removed, in other cells can be stay in the core of the formation of glycogen granules. And after that, students start the main enzyme, which is called glycogen synthase. You know from the first colloquium again that this enzyme is active in its dephosphorylated form. One more you know, this enzyme and all this process, which is called glycogen synthesis or glycogenesis, is activated during which state? Well-fed state, which means to start synthesis of glycogen in the liver or in the muscles, we need to have enough glucose right now. For the muscle tissue, it depends from the exercises. If there are physical exercises right now, which means we need rapid energy production, the glycogen synthesis won't be. But if there is just high glucose without exercises, of course the direction will be glycogen. And glycogen synthase start action by the adding of 1 by 1 glucose molecules, but from UDP glucose again. Don't forget UDP glucose. Again, UDP glucose is used. And there is formation only of 1,4-glycosidic bonds. But you know that glycogen is not a linear molecule, it's a branch molecule. That's why when are formed around 11 glucose molecules in one linear chain, the action of glycogen synthase will stop and start the next enzyme, which is the opposite of the branching enzyme, and it is called branching enzyme. This branching enzyme from non-reducing ends again catches these 6-7 glucose molecules and moves them to the contact them with alpha 1,6-glycosidic bonds, and now the branch is formed. That's all. After that, again, glycogen synthase 1, 2, 3, 4, 11 stops. Catches some 6-7 glucose molecules and moves them to form branch. That's all. That's all, students. And this is the formation of glycogen. In this scheme, you can see that all monosaccharides can be combined together to form glycogen. Here the question is it possible fructose to be converted to glycogen? Yes, yes it is. How? Fructose It depends from the organ, but if we're talking about the liver glycogen, fructokinase, then fructose 1-phosphate, aldolase, different trioses, and after that can be form GHAP up to form glucose 6-phosphate, then glucose 1, UDP glucose, and glycogen. If we're talking about the galactose, the same will be. Not the same exactly, but will be produced galactose 1-phosphate, then galactose glucose 1-phosphate, 6-phosphate, and again will be produced UDP glucose from glucose 1-phosphate directly, UDP glucose and glycogen. That's why, yes, all monosaccharides can be converted to glycogen. And actually, this is the main direction of monosaccharides, to become storage in the form of glycogen. A regulation. How many regulations you know about? Allosteric regulation and reversible covalent modification. Yes, these are used both of the regulation pathways. The main enzyme of glycogenolysis, glycogen phosphorylase. The main enzyme of glycogen synthesis, glycogen synthase. You know this scheme from the first colloquium again, but this one is more complex because there is some more information about the kinase reaction. The protein kinase reaction, phosphorylase kinase enzyme, exists in two forms again. In two forms, phosphorylated form and dephosphorylated form. And again we have protein kinase, which is used for the phosphorylation of phosphorylase kinase to become phosphorylated form and active. Actually, all enzymes in this scheme are active in their phosphorylated form. All of them. Phosphorylase kinase is active in phosphorylated form. Glycogen phosphorylase is active in phosphorylated form too. The dephosphorylation of this enzyme is thanks to phosphatase enzyme with one molecule of water again. And insulin activates both of the phosphatases, this one and this one. Both are activated by the insulin. Both of the kinases are activated by contra-insular hormones again. And this is the complex part of the scheme. All the rest, you know. Activation of glycogen phosphorylase, two main pathways. Yes, there are two pathways for the activation of glycogen phosphorylase, and the hormonal pathway, the hormonal regulation is the first one. This means that everything depends on the hormonal ratio, insulin-glucagon ratio. Initiation of glycogen breakdown depends on this ratio. If there is high level of adrenaline, the adrenaline action is mainly in the muscles. If there is high level of increased activity of glucagon, the liver glycogenolysis will be activated. There is one very big difference between liver and muscle glycogenolysis. What is, students? What is the difference? The difference is the final product. What is the final product in liver glycogenolysis? It's a free glucose. Why? Because of the presence of glucose 6-phosphatase. That's why liver glycogenolysis is used for the maintain the glucose in the blood only, but not the muscle tissue, as we said in the beginning of the lecture. Muscle tissue, the final product of muscle glycogenolysis is glucose 6-phosphate, but not free glucose. Yes. And in the liver, adrenaline can also, actually the action of glucagon or adrenaline is thanks to cyclic AMP pathway with protein kinase A and phosphorylation of the kinase. But there is one more pathway in the liver from adrenaline, another pathway, which is binding to alpha-2 adrenergic receptor and activation of one other pathway, not protein kinase A. This pathway is called activation of phosphorylated phospholipase C pathway and the formation of lipid mediators with increasing of intracellular calcium concentration and etc. You will see this when we talk about signal transduction pathways. The second pathway for the activation of glycogenolysis is not hormonal, but depends on energy. Energy dependent pathway. When ATP is low, this means no energy, which means high AMP. But AMP activates one AMP dependent protein kinase, which is called AMPPK. And this PK directly phosphorylates glycogen phosphorylase or stimulates phosphorylase kinase, which means activation of glycogenolysis thanks to information from energy balance in the cell, but not hormonal regulation. There is here. Calcium activates glycogen phosphorylase kinase, increase the calcium. Calcium is connected to troponin C, is bound to troponin C, and calcium activates this phosphorylase kinase. But after the activation, it is used for the phosphorylation of glycogen phosphorylase to become phosphorylated form, which means active. At the same time, phosphorylation of glycogen synthase will convert the enzyme to become inactive, because the active form of glycogen synthase is its dephosphorylated form. Why calcium is activator? Because it is used for the muscle contraction. That's why if there is high calcium, we need more energy, more glucose to produce energy for the muscle contraction. We need more ATP, that's why. And this is the regulation, the reversible covalent modification of glycogen synthase, the active form, dephosphorylated form, the enzyme. Which hormone activates glycogen synthase? Insulin. Why? Because insulin activates phosphoprotein phosphatase enzyme. Which hormone inhibits glycogen synthase? Contrainsular hormones. Why? Because they activate the protein kinases. That's why. And the regulation of glycogen synthase, there is one more activator, allosteric activator. It is the active form of glucose, the first active form of glucose, which is glucose 6-phosphate. If there is more glucose 6-phosphate, it activates glycogen synthase too, except the phosphorylation. And the last are diseases. Which are diseases? What is the name of these diseases? Where there is deficiency of any of the enzymes. They are called glycogen storage diseases or glycogenoses. And the defect organs can be these organs where there is huge amount of glycogen. Which are they? Liver or muscle tissues. Maybe the affected organ can be the liver, with enlargement of the liver and etc., or can be muscle tissue. It's important to know which isoenzyme is defective, the liver one or the muscle one. And let's see all glycogen storage diseases. In this table, you can see the enzyme deficiency of all these enzymes. If there is deficiency of glucose 6-phosphatase in the liver, the common name of this disease is von Gierke disease. And the glycogen structure is normal. If there is deficiency of enzyme alpha-1 for glycosidase, Pompe disease will be the name, and etc. All these diseases. We'll talk about the first one, Gierke disease. Now, clinical case. Von Gierke disease. The patient is a 4-month-old girl. For the past 14 days, the child became apathetic, had poor feeding, and showed signs of muscle weakness. She lost approximately 600 grams of body weight. The onset of high fever, likely due to an infectious disease, and tachypnea, rapid breathing due to oxygen deficiency, necessitated admission to a pediatric clinic. From physical examination, upon admission, the child was in a poor general condition, febrile 38.1 degrees Celsius with deep and rapid breathing. Episodic tachycardia, which means accelerated heart rate, was observed without evidence of cardiac abnormalities. On palpation, the liver was formed about 5 centimeters below the costal margin. Normally the liver is soft and not palpable. And let's see one moment. Okay, the table with laboratory tests. Glucose, hypoglycemia, severe hypoglycemia. pH of blood, acidosis. Lactate, that's why there is acidosis, lactic acidosis. The partial pressure of carbon dioxide, very low. Standard bicarbonate, very low. Serum triglycerides, you know that triglycerides means triacylglycerols, okay, the same. Triglycerides are very high, and urine ketones, ketone body in the urine, 3 pluses, the normal without. Discussion of laboratory results. The acute infection complicates the clinical picture. The symptoms are determined by acidosis because pH less than 7.4. What is the normal pH in the blood? Between 7. Yes, 4, 5, yes. But less than 7.4 is acidosis. Actually, a decrease in standard bicarbonate indicates metabolic acidosis, leading to tachypnea, this rapid breathing, with increased carbon dioxide exhalation, which means reduced partial pressure of carbon dioxide and depletion of bicarbonates. The presence of hypoglycemia and acidosis in a young child indicates impaired glucose homeostasis. Why? We said in the beginning of the lecture, how can we maintain the glucose in the blood? If except carbohydrate food, glycogenolysis and glycogenogenesis. That's why the reasons can be in both of the processes. These are the possible causes. Hepatomegaly, along with these symptoms, is a characteristic sign of impaired glycogen breakdown, which means the initial diagnosis is glycogen storage disease. But the question is, which enzyme is defective? And the initial interventions for hypoglycemia and acidosis is intravenous infusion of glucose and sodium bicarbonate to stabilize the condition. Diagnostic approach: collection of biopsy materials, examination of glycogen content. And let's go directly to the scheme, students, to see what is happened during the deficiency of this enzyme, which is called glucose 6-phosphatase. Okay, let's follow the reactions and discuss all symptoms. Where is the enzyme? Here is the enzyme. This enzyme is a part of actually glycogenogenesis, but glycogenolysis uses the same enzyme in the end of the glycogenolysis in the liver. Let's remind students what will be happen in any enzymopathies if there is any enzyme deficiency. What are the consequences? Number 1. Less product, and number 2, high substrate. These are the classical consequences of any enzyme deficiency. First, because of the less product, hypoglycemia will be. When we rely on this enzyme of glycogenolysis, especially early in the morning. Why? Because of the night sun. And in the morning we don't have any glucose in the blood because we don't eat during the night. That's why we rely on this enzyme early in the morning, because this is the end of liver glycogenolysis. And this is the explanation for the first symptom, hypoglycemia. The level of glucose in the blood was 2 millimole per liter, very severe hypoglycemia. The second consequence, we said increased level of substrate. The substrate is glucose 6-phosphate. But this enzyme is a key metabolite, key intermediate from many processes. Well, where will go this enzyme if it is increased? Maybe to pentose phosphate pathway to form pentoses. After that, synthesis and catabolism of purine nucleotides. Uric acid will be produced in the end, and hyperuricemia will be the second symptom, and the disease is called gout. Okay, the next. After the increasing of this glucose 6-phosphate can go to glycolysis process. But the glycolysis process after that, after the formation of this trioses, can go to glycolysis by glyceraldehyde 3-phosphate, pyruvate, and after that to alanine with hyperalaninuria, third symptom, or formation of lactate with lactic acidosis that we saw in the laboratory tests. The second direction is from GHAP to the right. GHAP, glycerol 3-phosphate, synthesis of TAC, and hyperlipidemia with high level of triglycerides in the blood. And hyperlipidemia will be not only from glycerol 3-phosphate, but also with the production of fatty acids from pyruvate and acetyl coenzyme A. And cholesterol level will be increased too. Why? Because of the increased level of acetyl coenzyme A at all. That's why both of them will be increased, triglycerols and cholesterol. All this is called hyperlipidemia. Okay, that's what about the treatment? Actually, this disease is hereditary disease, and the reason is where? It's not where is the reason? We said that there is deficiency of glucose 6-phosphatase, but where is the reason? In the gene. It's a genetic disease. In the gene is the reason. That's why the treatment will be symptomatically. To management the acute condition, correction of hypoglycemia, correction of metabolic acidosis, therapy of the infection, and support some about the food. What will be the diet, need to be the diet? With low carbohydrates, with low but frequent portions of carbohydrates. Yes, frequent small portions of carbohydrate containing food to prevent hypoglycemia. Yes, and continuous monitoring of blood glucose levels. And that's all from the first lecture today, students. Thank you.